In this presentation, we have to explain the two terminologies often used under genetics: penetrance and expressivity, concerned with the expressive ability of a gene or a genotype, whether it may be a normal or mutant genotype or the gene, how it expresses among the individuals in which it is present. Now you say there are 5 individuals in which this genotype present by some experimental process, survey process like a pedigree analysis, family tree analysis, and even by some crossing experiments. It's possible to say the genotype of a particular character in an individual. So by some survey method, you are able to say these 5 individuals possess the genotype for a particular trait. And you see that how many individuals express that trait, carry that trait. Now if you find all the 5 individuals show the trait to be expressed by that particular genotype known to be present, that way you can say that the penetrance is complete. That is, the expressive ability of the particular gene in all the individuals in which it is present. Then such kind of ability said to be complete penetrance. So if a gene is able to express its trait, its characteristic in every individual in which it is, then it may be called a complete penetrance. Now in this case you see the same 5 individual, you are sure that they possess the particular genotype, but only 3 individuals show the trait out of the genotype. Or in other words, you say this gene or the genotype able to express its characteristic only in 3 individuals out of the 5 individuals in which it is. So the 2 individuals not showing the trait of the particular genotype. Accordingly here the penetrance said to be incomplete. That is, not expressing in all the individuals. The genotype or the gene unable to express its trait in all the individuals in which it is. Even in percentage you can indicate. Out of 5, only 3 shows the expression of the particular genotype, and in 2 absence of the expression. Accordingly here the penetrance is 60%, because in 2 it is absent. But in the previous case you say in all the 5 there you can find the expression of the genotype. Accordingly here the penetrance is 100%. So that way also you can express the level of penetrance, the proportion of penetrance among the individuals in which a particular genotype present. And expressivity is also concerned with the expression of the trait among the individuals in which the genotype present. Anyway, there may be difference in the nature of the expression. Now here you see the genotype present in these 2 individuals and expressing its trait in both the individual without much difference. Accordingly you can say the equal expressivity or uniform expressivity. So the intensity of the expression or the degree of expression is identical in all the individuals in which a particular genotype present. If it is a disease causing gene or a mutant gene, you can measure it, you can indicate it in terms of the symptoms, whether the symptoms mentioned for a particular disease is completely expressed, all the symptoms present with these individual, in both the individuals. Then you can say the expressivity of the particular genotype is uniform. And in this case you say two individuals carrying the same genotype, but the expression shows some difference. So the variation in the expression, the level of expression, degree of expression. In one it is complete, or you say more number of symptoms expressed. And in another, even though it possesses the same genotype, the expression of the genotype seems to be very less. Only less number of symptoms are expressed. So in other way you say the intensity of expression is less, or degree of expression is less in this case. So this type of expression is said to be variable. But whether it is uniform expression or variable expression, you can say there is the penetrance. But to what extent it has penetrated? Accordingly, there come the important